So before beginning with the class, I think there are some confusion with like amongst everyone. So all those who are thinking that they have joined for pharmacovigilance or they have joined for clinical data management or CR, but the course structure, the module structure is designed in a way so that the basic concepts of pharmacology are reiterated and revised. Okay. And along with that, we focus on clinical research and clinical data management. Post that, the topic moves into PV and RA. Okay. That is how the course has been designed for everyone. Now, according to the courses that you have specifically opted for, maybe you have opted for clinical SAS. So in terms of clinical SAS, you will require a background of the clinical data that you are handling, right? Therefore, you will be given classes for clinical research, CDM, and then clinical SAS. Just before proceeding on to what you are analyzing or what you are working upon, you require the classes which will make you understand the kind of data that you are handling. Okay. I suppose, Sanjan, this has like resolved your query.
Yeah, ma'am.
And the next thing, the next thing is related to how our classes will be conducted. So it is scheduled for weekdays, right? Pharmacology is for 7 to 8. And
Your voice is breaking up.
Viswana, do you want to say something?
Yes, ma'am. Actually, your voice is breaking out. Ma'am.
Now is it better?
Yeah, yes, ma'am.
Okay. So I was saying that regarding how our classes will be conducted, pharmacology, the timings are from 7 to 8, right? These are scheduled for all weekdays, Monday to Friday. And in the end of it, like during from 28th July to 31st of July, I think 28th and 29th is a weekend. So in that scenario, this will be conducted during 26th and 27th. The 26th, 27th, and 30th and 31st of July, these 4 are your assessment days, wherein you will be completing, wherein you will be showing your PPT, PowerPoint presentation. You have to develop a maximum of 10 slides or a minimum of 8 slides of presentation. And it should be from the topics that we are discussing along the course. It doesn't necessarily have to be a reiteration of the topic. You can select the topic and from there you can discuss upon the recent research which is going on those concepts or modules. Okay, that also works fine for me. Then you will be required to give a MCQ questionnaire, and along with that, not long, but subjective questions, worth like maximum 2 to 3 subjective questions. Okay. And for this, you will be given a time of 1 hour each. Like when I will be taking the slides, it will be on 26 and 27 that we will be focusing on slides, and on 30 and 31st, you will be required to give the MCQ questionnaire on one day, and for the second day, your long answer question will be scheduled. Okay. And for this entire scenario, it is indispensable that all of you will turn on your cameras. Okay. You cannot turn off your camera and write the exam. And another reason that I'm mentioning it way before we complete the syllabus is because I do not want anyone say on that day that I'm traveling or I am in a family function or there is some other reason because of which I cannot turn on the camera. That won't be entertained and you will be given 0 for that particular exam, whichever day you fail to turn on your camera and complete the exam accordingly. Okay. And another kind of an interactive session. See, basically another way that you will be graded for your for this particular module of pharmacology is how much attentive you are towards the class. It doesn't necessarily focus on your 100% attendance, but yeah, that attendance should be complete. It should be your conscious attention with the class, and you must be interacting with the class. It should not go on like you will keep yourself on mute and never say a word if you are being asked a question or if any question is being asked on to the open audience, right? So it is a must that you all should interact. Also, We will do some mock sessions, mock interview sessions from the topics that we are covering in pharmacology, so that I know that there are some students, those who are already from pharma background, but there are also some those who are absolutely not from pharma background, are from either general sciences or chemistry or biology background. So it will be a great leverage for them in order to, like, when they are providing the answers in the class, if we conduct such mock sessions, it will be a learning experience for both the parties, like for the interviewer and also for the interviewee, right? So that is how our 1 month long course is designed. Okay. I suppose now there is no confusion regarding how the exam will be conducted and all. Tanjan, this was it, right? You wanted to know regarding these scenarios only, right?
Yeah, ma'am.
Okay. Then we shall let's proceed with the class. So yesterday,
Ma'am, and
Ma'am,
yes,
hello,
please. Yeah, Tanjan,
ma'am,
go
and
ahead.
totally, how many modules are there in pharmacovigilance and CDM, ma'am,
In
particularly?
pharmacology.
In pharmacovigilance, in the sense like you will do first pharmacology, and then you said like you'll go for that pharmacovigilance, right?
I won't be teaching you pharmacovigilance. Pharmacovigilance will be taught by another person who is
Yeah,
responsible.
yeah,
Yeah.
got
So
it. But
I
ma'am,
am not
I just
sure. I'm not sure about the total number of modules in PV. I can tell you
mm,
about CDM and regulatory affairs, because those are the other two subjects that I take classes for. So I can
mm,
tell you about that, but I cannot be completely sure of the total number of modules that are there in pharmacovigilance, right?
yeah, sure, ma'am. Okay, thank you.
hello,
Um,
hello, ma'am.
yeah, your name is Sushri Ankita, right?
Yes, ma'am.
Okay, yes.
Ma'am, what has been taught yesterday? I have not attended the class.
I don't think that you attended even Monday's class.
Sir, ma'am, I have not joined in the group Monday. I joined yesterday.
Okay, but okay, so you couldn't attend yesterday's class as well, right?
Yeah.
So, Shishri Ankita, we have started with the basic introduction towards, I want to understand what is your educational background, Ankita?
I have done MSc in chemistry.
Yes, in chemistry. Okay.
Yeah.
Well, we have introduced what pharmacology is, okay, to the class. And considering that we have given some 2 long days only to the basic introduction of pharmacology, I will request one of your classmates to please come ahead and explain what pharmacology is. So we will begin, we will do it one by one. Yeah, so Ziaul is the first person to volunteer. Ziaul, please help the class in recollecting the basics of pharmacology.
Yes, ma'am. Good evening, everyone. Here, pharmacology, the word pharmacology is derived from a Latin word, where is pharma. Pharmacon means making drug, and cology means study about the drug. So what we'll study here, pharmacology, it is a branch of science which will deal with the effect of drug in the body. Means what will be the mechanism of the drug inside the body and how it will affect the normal physiology of the body. Am I clear?
Yes, Ziaul. But Shushri, were you able to understand what Ziaul said regarding the basic concept of pharmacology?
Yeah.
Okay. Okay, thank you,
And
Ziaul. You want to add something?
yes, ma'am. We can say that pharmacology also deals with before, if we are understanding pharmacokinetic and pharmacodynamic all together known as pharmacology. In pharmacokinetic, we'll be studying ADME property, that is absorption, distribution, metabolism, and excretion, as well as that is means when a drug is taken inside the body, what body does to the drug. That is pharmacokinetic. And when the drug is already gone to the receptors, what drug does to the body. If a person is getting ill, how the drug is getting making the person cure from that disease. What will be the mechanism, the whole mechanism of the drug, and the response to means against our disease. That's whole collectively can be called as pharmacology. And we'll be studying here the adverse effects also of the drug, side effects also, beneficial effect, and means risk factors of the drug also.
Absolutely, absolutely. Thank you, Ziaul. Thank you.
Yes,
Uh,
ma'am. I have one
yes.
doubt. How it is differ from pharmacovigilance? Also, pharmacovigilance deals with the drugs effect and
Can
Okay.
I
Pharmacology. Yes, Ziaul, please proceed.
I said that it will give a knowledge about the body's normal physiological condition and when the drug is taken into the body, and it will say all the side effects and all. We are now when you are going for pharmacovigilance, it is a phase of the clinical trial. We know in the clinical trial, phase 1, phase 2, phase 3. Up to phase 3, then we'll get marketing approval, and phase 4 is after marketing. We have to see. Now, adverse effect is linked with pharmacology only. If you are not knowing the mechanism of the drug, if you are not having the knowledge about the mechanism of a drug and how it is getting affected, then means we will not get the pharmacology, because we know pharmacology is mainly to monitor the adverse effect of the drug. So, adverse effect is a part of is a part of is a part of drug development process, and that adverse effect can be known if we are having good knowledge of pharmacology. Am I clear?
Absolutely.
yes,
Okay, I will clarify it also. So, Shri, as
Yes.
Ziaul very correctly said, that pharmacology, if you break down this word into its root derivatives, it is pharmacon and logy, right? So pharmacon is the drug, like it is a name given to drug or any substance which can potentially alter, which can potentially enhance, improvise the physiological working of the body. Okay. Then logy is the study of this pharmacon. So the study of drugs, that is pharmacology. Pharmacovigilance, if you break down this word, it is pharma with that of vigilance, compliance and vigilance. What does vigilance, if I just put out the word vigilance, what is the literal meaning of vigilance? What do you think? As a common person, you don't have to go into the deep knowledge of pharmacy and pharmaceutics, but yeah, just the term vigilance, what do you think it is?
monitor.
Monitor.
Monitor, absolutely.
Yes.
Like most simplest explanation for vigilance is like surveillance is, I would rather say it is more of an industrial word still. But for a complete fresher, if I would have to explain the difference between pharmacology and pharmacovigilance, is that vigilance is about monitoring, is about looking after, right? Looking after what are we looking after? So the kind of adverse impacts that can be caused by this drug, we are looking after the possibilities and probabilities of these ADRs which can be caused upon consumption of this drug, right? But that is very specific to encountering and reporting those ADRs. Okay. But
Yes.
pharmacology is a broad subject. It is a broad subject which includes in itself a deep, or rather, what should I say? It includes in itself a long drawn procedure of understanding like the understanding begins from the research that we perform for extracting the compound, which is actually responsible for providing us with beneficial, like therapeutically beneficial impacts. Okay.
Yes.
Now, this particular substance, it is referred to as API. Have you heard of the term API?
Yes, ma'am. I have heard the term maybe, but I don't know the full form.
And what is the full form?
Active pharmaceutical ingredient.
Yeah,
pharmaceutical-
Charita, please be a bit louder.
Active pharmaceutical ingredient.
Absolutely. So Shri, API is active pharmaceutical ingredient. Okay. Now, pharmacology as a domain, as a stream of education that we gain, it starts from the very point that the research begins for the API. Okay. And the moment we have extracted this active pharmaceutical ingredient, we start off with our research for how this drug will perform when consumed. Okay. And not at this moment, it is not a drug. At this moment, it is just a chemical compound. Okay. The first line of action, the first line of action in terms of pharmacology is to understand what are the different additives or the different excipients with which this API can be mixed and added in order to formulate a drug in the first place. Okay. So that is the first thing, that we are checking about its stability, its chemical structural stability in the presence or on bonding with other drugs not drugs, but on bonding with other chemicals. What is its stability and how much is it compatible with those excipients? So that study, from there we begin with the evaluative characterization in terms of pharmacology. After that, once the drug has been formulated, we have to determine what is the ideal dosage of this drug for certain conditions, right? Because you must have observed on a daily basis that one particular drug is provided in different kind of dosages. For some patients, it may be 250 mg. For some patients, it may be 500 mg. For some patients, it is strictly advised for only consuming it in the morning, right? So for some patients, it is provided for twice a day, right? So this characterization, that what will be the dosage, what will be the frequency of the dosage, what will be the total number of period for which this particular drug can be administered to a human body or an animal body. Okay. So this characterization, this evaluation, this is the second activity that we are performing in terms of pharmacology. Then we come down to study the effect of pharmacokinetics and pharmacodynamic. I think you must have heard these two terms when Ziaul mentioned it, right?
Yes.
So he also explained what is pharmacokinetics and what is pharmacodynamics. Do you remember his words?
Yes, ma'am.
Okay. So Sushri, let us hear from Rizwana. She will reiterate, and it will also help recollect the other classmates also. Rizwana, please mention what is pharmacokinetics. Okay.
Ma'am, pharmacokinetics is about the study of how our drug does, how our body does to the drug. It's like our movement of drug, and
Absolutely. Thank you, Rizwana. Can I have someone else to please explain what is pharmacodynamics?
Hello, ma'am.
What drug
Yes,
does to the body?
Joshna. Okay, Charita. Charita and Joshna wants to explain, right? So we will give Joshna a chance this time because you answered one thing before this. Okay, Charita?
Okay, ma'am.
So when the drug reaches the systemic circulation or the target organ, it shows the action, it shows the activity.
Absolutely. So the kind of activity which is exhibited by this drug when it is inside the body, that is what pharmacodynamics is, right? So Shushri, I suppose now you have understood the difference in between pharmacokinetics and in between pharmacodynamics.
Yes, ma'am.
Right. So pharmacokinetics is what? Pharmacokinetics is the study of how our body is reacting to a certain drug, right? And pharmacodynamics is what the drug is doing to our body. Okay.
Yes. Yes.
Okay. So till this point, Ziaul had already explained. Okay. Considering that he has already brought up what are the different the proper name to define this is the different branches in pharmacology. So I will directly go over there. Okay. Okay. And one thing that we are missing, we have missed out in terms of all what that we read for in pharmacology is the science of drug development. that I explained in terms of the characterization of the research procedure that is there for extracting the active pharmaceutical ingredient, right? And the journey from that API to ultimately becoming a drug, which is the commercial form of the drug that we are consuming. Okay, so this entire procedure, it starts from the research of the API, then it moves on to drug formulation, um, uh, characterizing the stability, the physicochemical characteristics of excipients and the compatibility with this API, then ultimately formulating the drug, listing out the ambient condition in which it should be stored, how it should be consumed, then moving on with the process of clinical trial as an IND, right? And then ultimately listing this product or getting your IND approved by the regulatory authorities for it to be commercially produced as an NDA, right? So once and until the point that the drug is in the market, it is being consumed, right? So this entire scenario of drug development, that also is a broader part of pharmacology only. Okay. See, we have given different names. We have given these names of clinical trials, clinical data management, pharmacovigilance, regulatory affairs, quality control, quality assurance. But tell me one thing, are they doing anything which is different from the concepts that we are learning in pharmacology? No, right? It is just stepping up with the essence of pharmacology. It is about differentiating these different segments into certain branches so that like we can say rather that it is more of a naming convention that we are putting out in order to define the departments or in order to define the category of work that is being done at those certain steps. Okay. And yes, it obviously may come up as a question that how exactly, because I'm saying that in pharmacology also we are studying the adverse impacts. We are studying how drug interaction or drug interaction with that of the body can cause certain adverse reactions, adverse events, which is harmful for the body. And if it is not causing adverse impact to the body directly, then what are the underlying factors which can cause harm to the body? So when if this entire thing is being studied under pharmacology only, then what is the need of pharmacovigilance? In pharmacology, we are studying these. We are characterizing this out. We are researching on this. We are like developing our knowledge base, developing a database for this entire literature of knowledge. But pharmacovigilance is about characterizing this information, reporting it to the regulatory authorities, and ultimately communicating it to the general public. That is what pharmacovigilance is meant for. Because if you can recollect, I suppose all those who are from pharma backgrounds, they must have went through this complete module at least during their bachelor's, that what were the different scenarios earlier during the 1800s or 1900s, when there was no proper like convention, no proper pharmacopoeia, no proper quality assurance or quality control team. And in those times, the kind of vaccines, medicines that were manufactured or produced, so they were actually responsible, like not the disease, not the causative organism, but the medicine which was taken for betterment of that disease was actually responsible for causing death to the patient. So in order to avoid those scenarios, we have focused upon creating departments, dedicated departments, which can help in assessing and evaluating the different facilities, understanding what is the QA/QC scenario of these things. And also, even if the QA/QC is absolutely prim and proper, but ultimately, is the API good enough for human intake or animal intake? So in order to determine that, and also to let know of the general public that what should be consumed and what should not be consumed, we have created these dedicated departments referred to as regulatory affairs. And underneath, we have certain sub departments which is pharmacovigilance, material vigilance, post-market surveillance. Okay. Okay. So Shri, now I
Okay,
suppose
Yes, yes, ma'am.
you could understand the difference.
Yes, ma'am. Ma'am, that means pharmacology is the study of everything, and if we then
Branch
classify,
out. Yes,
yes,
yes, absolutely.
pharmacovigilance is one of that part, and everything is one of that part of pharmacology.
Yes, yes. Everything is a part of pharmacology. Everything is
Yes.
not a part of pharmacovigilance. Everything
ma'am.
is a part of pharmacology.
Yes,
Yes, we can
pharmacovigilance is one of part of the pharmacology.
absolutely. Yes, we can absolutely say that pharmacovigilance is a part of pharmacology.
Yes, thank you.
We can say, like, in the most layman terms, if I have to say, pharmacology is India, and the different states are the different branches over there in a country. Okay, that is how
Yes.
you can understand the different branches. So then, like, now when we are understanding API, we come down to a scenario wherein we understand that yes, this pharmaceutical ingredient, this chemical compound is ultimately being extracted from certain sources, right? This I think we are all aware that ultimately we are sourcing the pharmaceutical compound from certain. Yes, please go ahead.
Ma'am, your voice is breaking, ma'am.
Is it good now?
Yeah, it's good now, ma'am.
1 second, just give me 1 second. Is it better now?
Yes, ma'am.
Can you all hear
Yes,
me?
ma'am. Yes.
Yes, ma'am.
Yes, ma'am.
Okay. So now we were discussing regarding the different sources, right? So if I have to classify, there are animal sources, and then there are mineral sources, microbiological sources, and also synthetic sources, right? So in terms of the natural sources from where drugs are obtained, plant sources and animal sources are the most, what should I say, like it is the like first classification that we perform in terms of resources, and also the most ancient form of medicine that we have, right? So in terms of plant sources, like maximum drugs that we are aware of, they are they belong to the plant sources. At this moment now, we have modern technology, we have genetic engineering setups which help us in what should I say, in collaborating the plant source with that of a microorganism so as to improve or enhance the effect of that drug. But in if we think about the primitive scenario, we can say that plant source is the oldest drug source, right? And maximum parts of the plant, whether it is the leaf of the plant, stem of the plant, fruits, roots, whatever, they can be like through complete chemical processing and microbiological procedures, we can extract the pharmacologically active and potent compound, right? Which is ultimately responsible for producing certain chemical compounds, which is quite helpful in treating various ailments. Some of the common resources that we are aware of, I suppose everyone has heard about the drug atropine, right? At least pharma people must have heard about atropine.
Yes, ma'am. Yes, ma'am.
Are you aware which plant source we are obtaining atropine from?
Yes, ma'am. Narcotics drug.
Atropine is being obtained from
Atropa belladonna.
Atropa
guess,
belladonna.
ma'am.
Very absolutely correct. And even there are homeopathy medicines as well, which is extracted from this plant. Okay. Then another source is that cardiac glycosides. I think we must have heard about tablets known as digitoxin or digoxin.
Yes, ma'am.
This is a cardiac glycoside, and these this is extracted from the leaves of a plant named as Digitalis
Digitalis.
purpurea. Yes, so that is how we are gaining the cardiac glycosides. Then eucalyptus is one of the common source of drug, and eucalyptus oil, like leaves. Eucalyptus oil is basically extracted from the leaves of the eucalyptus tree, and it is you if you go through the label, it is one of the most important compounds for treating ailments like cough. So cough syrups have uh, eucalyptus oil as one of the common sources, and that is again obtained from the leaves of eucalyptus. So that is about the leaves. Then there are certain flowers also, which is helpful in providing us with certain drug sources. Like we have um, morphine. Morphine, we can extract morphine from poppy plants. Okay. Then if we have to talk about the fruit as a resource for extracting API, then I think there are some medicines which are used for treating constipation, which is extracted from certain fruits of plants. Okay, then we have for if we think of leaves, then I suppose many of us are aware of castor oil, right? So castor oil is again obtained from the leaves. The extraction of leaves are performed for castor oil. So that is how like how vivid how vivid plants are in terms of providing us with certain chemicals, certain potent chemicals. Then we come down to the second source. The second source is the animal source. So can any one of you name me one of the most common drugs or the most common form of drugs which was like till a very long period of time? I would say like even till the last decade, we have extracted this particular drug from pigs. Are you aware of this
Insulin, ma'am.
drug?
Insulin.
Insulin, absolutely. So
Fixed flavor.
yes, so then we have um
Liver cod oil, ma'am.
hello.
Liver cod oil.
Yeah, will you please repeat?
Cod oil, ma'am. For
Yes,
vitamin A.
yes, absolutely. That is one of the most commonly used medicines that we consume even till date, right? Then okay, so one of the sources of thyroxine, okay, is from sheep thyroid. Okay, and this particular medicine is most often given during patients with hypertension. Okay, then there's a particular hormone which is extracted from the urine of pregnant women. Okay, and this is human chorionic gonadotropin. So this HCG, this is used for the treatment of infertility. That again is a animal source, right? Um, Moving forward, this is more about the plants and animal sources. Then again, we have mineral resources, right? So one of the common ailments which is treated by the mineral resources, I suppose everyone is aware about anemia,
Yes, ma'am.
right?
Iron.
Yes. So anemia is one of the common ailments which is actually treated by a drug which is
Iron.
from a mineral source, right?
Yes, ma'am.
Syphilis is treated by mercurial salts. Um, Then again, zinc, zinc oxide is one of the most commonly used and um, chemical constituent in order to treat wounds, in order to treat eczema, and even certain skin conditions are actually treated by zinc oxide paste, right? So then again, that is what we are extracting from a mineral resource. Zinc is also provided to us in the form of supplement, right? Zinc supplements.
Yes.
We have another commonly used antiseptic, which is
Potassium ion.
mint, right?
Yes.
Like you were giving some other example. Please go ahead.
Potassium iodide, ma'am.
Yes, absolutely, absolutely. That again is a mineral resource for drugs.
Yes.
Okay, I didn't see the message earlier, Reshma. Yeah, thank you. So Shri, another help from your classmate in order to differentiate further pharmacology and pharmacovigilance is in the chat box, and Reshma has correctly put out that the mechanism or the method of working for pharmacovigilance is to keep a watch on the API, on the active pharmaceutical ingredients. Okay, you can check that out. Thank you, Reshma. Okay.
Yes, ma'am.
We have one of the most common ailments that we find maximum population suffering from is rheumatoid arthritis, right? So there are some research papers which explain about the usage of gold salts, okay? Gold salt in order to treat rheumatoid arthritis, because we are aware that rheumatoid arthritis is that particular disease which I won't say disease, rather it's a condition in which is largely curable. Like it is the curable rate of this particular condition isn't that much successful, but gold salts has shown prominent results in terms of treating rheumatoid arthritis. Okay. Other than that, when we talk about synthetic or semi-synthetic sources, then basically the explanation of these synthetic sources is that the nucleus of the API, okay, that this nucleus of the natural source which is being used to extract the API, as well as the chemical structure of like how exactly that is altered. Okay, so we can say that the API which was being extracted naturally, we are treating it with certain chemical agents. We are treating it with certain chemical agents or certain microorganism sources in order to alter its structural composition. Okay, and ultimately the mechanical intervention that is being performed in order to create such medicines leads us to the formation of synthetic or semi-synthetic sources, right? Then we have microbiological resources. I think everyone is aware about the most famous microbiological source of drug, which is Penicillium notatum, right?
Yes, ma'am.
Yes, ma'am.
It does penicillin. Then we have streptomycin. Streptomycin, I guess everyone has heard of this particular drug. Also, like I'm talking
Yes.
also from the
Yes,
non-pharma.
ma'am.
Yes, ma'am.
Yes, ma'am. Yes, ma'am.
So from where is streptomycin extracted? From which microbiological organization?
Streptococcus.
Okay, like can you give me the full name?
Streptococcus.
Cerevisiae, ma'am. I guess like
Streptomyces gracious.
Okay,
Streptomyces gracious.
streptomycin is obtained from, yes, Streptomyces gracious, absolutely. And can you tell me for which particular ailment is streptomycin generally um, administered or prescribed?
Throat
That is
shorting.
tuberculosis, ma'am.
Throat shorting.
tuberculosis. And in terms of tuberculosis, if we have to explain, streptomycin is actually the second line drug for curating multi-drug resistant tuberculosis, right? Okay. Then we have certain other aminoglycosides as well, okay, such as gentamicin, which is again obtained from some species of streptomyces. Okay, then that is about the microbiological sources. Further, we have the recombinant DNA technology. I suppose people from biotech microbiology background, if anyone is here from there, from these backgrounds, they are quite familiar with recombinant DNA technology.
We also read, ma'am. Now
Oh,
insulin
okay.
is synthesized by that.
Yes, humulin, right?
Yes, ma'am.
Yeah,
Yes, ma'am.
sorry. yeah, I know that pharma people are obviously they must be aware, because this is again a part of part of pharmacology only, and ultimately we are shifting towards the trend of focusing more on the genetic engineering aspects for devising drugs, because it ultimately helps in treating out the root cause in an effective manner. That is there. And also another thing about these synthetic preparations or genetic engineering resources is that it cuts out on some of the adverse impacts which was earlier possessed by
Big insulin.
yes, absolutely. So recombinant DNA technology, ultimately what we are doing in this particular scenario, is there anyone who can explain a bit about recombinant DNA technology?
Human insulin is taken and plasmid of E. coli, there will be recombinant, then sulfur dibond will be like purified and
Absolutely. So basically, why we are mentioning at
what have they will become.
the name recombinant DNA itself gives out that over here we are altering the DNA structure, right? And what we are doing is that we are basically cutting out the DNA, okay? And this cutting out or cleavage of DNA is performed by the enzyme restriction endonuclease, okay? And this when this gene
Yes,
is when the DNA is cleavaged, or when it is cut out, so at that time we introduce the desired gene into the other segment, okay? And that is introduced with the help or via a viral resource, a viral organism, a bacterial source, and mainly in genetic engineering labs, the resource which is used for this introduction of the desired gene into the cutout gene segment is through a plasmid, right? So
yes,
this ultimately creates a new genetic combination, and this genetic combination is further inserted into bacterial cultures. When it is inserted into
yes,
bacterial, it is there, it is kept there for further growth, and it leads to an yield of a vast amount of genetic material which is further used for gene therapy, right? One of the major advantage that recombinant DNA technology provides is that huge amount of drug can be produced from one single batch, right? The drug which is obtained, or the the gene, considering that we are all introducing the desired gene, we are basically cutting out on any unwanted or undesirable chemicals, undesirable segment of the DNA. Therefore, the drug which we are obtaining is also in its most purest form, right? Therefore, we can say that yes, recombinant DNA technology, like the reason why it is gaining momentum these days and why maximum labs are focusing on this particular technology for further development and introduction of different drugs that are there, right? Okay. So this was about the different microbiological sources. Okay. Then there are certain Nobel laureates whom whose history has been mentioned over here. I will request some of you to please quickly, you can read this from the PDF from your LMS. So one of the major things, one of the major takeaways from reading these different Nobel laureates is, I would say, about Paracelsus, because this person is responsible for providing us with the concept of with this dose response concept. So ultimately, whatever drug that we are consuming, right? So even if we are consuming a drug which is paracetamol, we are aware that it has the least adverse impacts and the least side effects. But is it can we say about paracetamol that it won't lead to any adverse impact howsoever it is consumed? We cannot say, right? We can never assure a person that even if you are consuming 3 to 4 paracetamols in a day, and if you are consistent with this practice of yours, it won't lead to any adverse impact. We cannot say that, right? Because ultimately, till a certain dosage, a particular API, a particular drug will be considered as therapeutically beneficial. But beyond that, it won't be considered therapeutically beneficial, or rather we cannot even say that it will be considered as like a medicinal product at all, right? Because we are even aware of, for example, yeah, so we have all heard about drug addiction, right? We do not let our youngsters go into, like, get into the addiction of weed, right? Hemp, which is the scientific name is cannabis and more locally named as
Marijuana.
gaja,
Marijuana.
right? Marijuana. Now tell me, have you heard about some research papers describing, and not just research papers, there are some active clinical trials which is being conducted in the US for using marijuana for the treatment of cancer. Have you heard about this or not?
Yes, ma'am. It is legalized
Yes.
also.
Legalized in US, right? But
Yes, ma'am.
ultimately, it is something which leads to addictions and severe addictions, which can cause a person go insane to a point, right? But then that is again being used for treating cancer. Cancer, we are all aware that it is one of the most invigorating and infuriating diseases of all, right? It can relapse, it can rebound, and it will come back in various in multiple organ failures and all. But still, the choice of medicine that is actually being selected upon for treating this cancer, and also I must tell you that it is actually showing great results, right? It is showing potential results with 0% relapse, right? All those
Yes.
who have the habit of reading through the papers must have read about this news. So that is how we determine till when or at which quantity, at which dosage value will a particular chemical compound, will a particular plant source, animal source, whatever it be, will be conferred as a medicine, and beyond which it will turn as poison. So this person is responsible for providing us with that background, wherein we started to understand that everything which we are consuming as a therapy has to be bound in a certain quantity, and also about the frequency of events at which it can be administered. Okay. There are some other descriptions also regarding various celebrated pharmacologists and chemists who are responsible for providing us with various resources. Okay. I think you all can read through these different historical contexts. There is nothing new over here. How what we will begin now is about the different divisions, right? So what initially we were discussing regarding the different aspects of pharmacology. So we mentioned that it consists of pharmacokinetics, pharmacodynamics, right? But beyond that also, we can categorize it into certain segments, okay, which is inclusive of the different ways how a drug is impacting the body. Okay, for example, we can mention about clinical pharmacology, wherein we are very specific about the about about the aspect, about the scientific study of the drugs, okay, and also the simultaneous or concurrent investigations related to its pharmacokinetics and pharmacodynamics. Uh, Clinical pharmacology is the branch from where we derive out the different surveillance programs and vigilance programs that are being conducted today. Okay. The main objective for conferring or categorizing a particular branch as clinical pharmacology is to we can say is to enhance and improve the effect of drugs, improve the therapeutic value that a drug can potentially provide, and also the ultimate objective with which we are studying clinical pharmacology is to promote safety in terms of prescribing a medicine or in terms of consuming a medicine. Okay, then we have toxicology, chemotherapy, pharmacognosy. I suppose all those people, those who are from pharma background, are quite aware of what pharmacognosy is, right? So can anyone please help us in understanding what is pharmacognosy? Can I have a
Yes,
volunteer?
ma'am. A branch of science which deals with the means source of medication that is means obtained from natural sources like plants and animals, ma'am.
Right. And in this pharmacognosy, like the major objective is to identify the drugs, right? Either
Yes, ma'am.
or how they appear. That is how we are identifying the drugs. So this entire branch of pharmacognosy is to identify, and the reason we must identify, we must know how to identify a drug, is to avoid any chance of improper usage, right? So again, that is about pharmacognosy. Then we have pharmacoeconomics as well. So pharmacoeconomics is about the entire parameters which is related to the entire monetary parameters, economic parameters related to the drug development process, and also how much we can, the reason we go ahead and have this dedicated branch of pharmacoeconomics is because we have to, the population at this moment is huge, and we have to provide drug to a huge number of people. Okay. And if one particular batch of drug will cost us, say for, just hypothetically, if I would say that if one batch is costing a company approximately, in INR, if I have to specify, approximately 1.5 lakh rupees for one batch, then we can say that this is like very expensive. This will lead to expensive drugs. But we have to cut down there, right? Because our population, maximum population of the world cannot afford that much of expensive drugs. And not just India. I mean, cut out India. At least in India, you have the feasibility of like collecting drugs over the counter as well. However, if you go to America or UK, the possibility of collecting OTC is absolutely nil. And the cost of drugs, the cost of the medical therapy is really high. It is reaching sky high prices. So therefore, we have to understand what are the sources on which we can cut out on what, or how we can optimize this complete process of dealing with the complete drug development process. That is why we have this dedicated branch for pharmacoeconomics. Then we also discuss about pharmacogenetics and pharmacogenomics. Okay. The reason because we are going out at a stage wherein we are very specifically dealing with gene therapies. There are various autoimmune disorders and even certain cancer types which actually has a relation to the gene, right? So we have to focus on actually working on or correcting the alterations which has been caused to the gene, correcting out the mutation which has been caused to the gene. Now, yes, the success ratio of this process is kind of debatable, but we do have this separate branch which is improving each day in order to provide better medication and better therapy to those diseases as well. Then the last thing that we discuss about is pharmacoepidemiology. Pharmacoepidemiology is the study of the effect of drugs on the large population. For example, recently we had COVID, right? So COVID-19, we cannot say that it is over now. It isn't, because if you check, there are various comorbidities that COVID-19 has actually caused. There are many cases with people, those who suffered with COVID. Okay, and I'm talking about young people from the age group of 2021 to that of 40. All the people, those who fall under this particular age bracket and they have suffered from COVID, they are actually facing, particularly females, they are facing this problem of fluid retention in the body, particularly in our joints, okay, like knees, ankles. There are even various papers. So this complete evaluative study which is being performed in order to affect the effect of diseases, effect of drugs on patients, on the like general human population. That is the that is what pharmacoepidemiology is. And I would request all of you to please complete your reading through this chapter from your LMS, because there's nothing new which has to be explained. We have already discussed about the different parameters which are there. From tomorrow, we will begin with the second module. Okay.